The future of Canadian food is strong, and FCC is ready to help the next generation of innovators like you with financing tailored to your needs and resources to help you keep growing. To get started, visit fcc.ca/youngfarmer. Thanks to our show sponsors, the Wheat School, Profitable Practices, and MNP. Rooted in agriculture, MNP Ag Advisors combine field-level agronomic knowledge with financial and operational insight to help farm businesses make confident decisions. From the field to long-term growth planning, MNP turns insight into action. Learn more at mnp.ca.
Well, good evening, everyone, and welcome to the Agronomists. I'm Peter Johnson. I'm sitting in for Lindsey tonight. She unfortunately isn't able to host, so you have to put up with me, but I'm really ever so excited about our session tonight. We are going to talk about biologicals, some new data that has come out, as well as, what are the opportunities out there and what do we need to look for in terms of finding that magic silver bullet that we keep hoping biologicals will turn into. And I've got two excellent guests here with me tonight. At the end of a glorious sunny day, everybody, it looks a little bit like in the chat, we're thin on Ontario people because we are now an hour earlier. We sprung ahead last night with daylight savings time. So I guess all the Ontario folks are have already gone to bed. Perhaps they're just that tired. But let's bring in our guest, Greg Stewart from Syngenta. He's the biological specialist with Syngenta, and Dr. Laura Lindsey from The Ohio State University. It pains me, Laura, to have you with us just because your Twitter handle is Step Up
I know.
Soy. And you are the small grains and soybean specialist. So, anyway, tell us a little bit more, Laura, about what you do and what it's like in Ohio right now.
Sure. I'm the soybean agronomist for soybean and small grain, which in Ohio is soft red winter wheat. But we do have 5 million acres of soybean in Ohio and about 500,000 acres of wheat. So that's kind of the step up soy. But I love wheat. It's a great crop.
Yeah, redeeming yourself very nicely there, Laura. Yeah, absolutely. Greg?
How you doing?
Yeah.
I love wheat.
So,
I love wheat too. I like to eat bread and donuts and
you just don't like to grow it so much, right, Greg?
no, it's nice on the headlands. That's my goal for my next 10 years of my career is to move wheat just to the headlands.
And that's because you don't want to tramp corn on the headlands, correct?
I don't want to tramp the corn. I just want to tramp the wheat.
Yeah. And the soybeans, if I remember correctly.
Yeah, maybe once in a while some soybeans, but they can handle it.
Yeah, and so if people have seen Greg's presentations lately, he likes to show his one field where he's convinced a farmer to grow soybeans wheat rotation on the headlands and grow corn in the best part of the field, so the corn looks like it's a decent crop and the other ones don't. But yeah. And you aren't missing a Habs game tonight, Greg?
What is Monday? No, I don't think so. We're good.
But
we're good. We're hanging in
otherwise,
there.
Otherwise we wouldn't get you here. So let's talk about biologicals. And so I think right out the gate, Greg, let's go to and I know you said that you're not convinced that they're everybody's categories of biologicals, but whenever you've given a presentation on biologicals, I think you've done a nice job of kind of splitting them out. So before we get into what might work and what data we have on different products, just run through what those 4 different categories are.
Yeah. Well, again, like we've chatted in the past, I divide them out a little bit into categories that aren't always agreed upon by everybody. But one place to start is to suggest that there are 4 categories, and those 4 categories, for me, I put them in biostimulants. But when I put them in biostimulants, I put them in plant extract, mostly biostimulants. And so therefore, my next category would be biostimulants, but it would be all microbe based. So living organism biostimulants as opposed to plant extract biostimulants. And then sometimes I go to PGRs as my 3rd category in this group. And then I go to almost a separate category, which would be the humic acids. But again, some people would put all 4 of my categories into one category and they'd call it biostimulants. But I think for our discussion tonight, we get started maybe on breaking them out into plant extract versus microbe versus PGR versus humic acid. And of course, there's some other possibilities. Like I haven't even talked about endophytes that fix nitrogen. So that would be my 5th category. But that's the way I break them out. And I think maybe it's not a bad place tonight to start at.
Yeah, absolutely. And endophytes certainly would be another one that there's been lots of effort put into, but not as many positives so far as what you might hope, right, Greg?
Yeah. And you could obviously have put endophytes into my biostimulant microbe category, but because I love them so much, I moved them into their category of their own.
No, very cool. And so, Laura, you have done a bunch of and actually not just yourself, but the whole soybean or the whole group of soybean researchers across the US have kind of gathered together. And so just walk us through, if you could, a little bit of what you've looked at and why you chose the products that you did. And I think it fits right into Greg's first category, right? Second category, the biostimulants microbes. So just, it's a complex area. What did you do and what did you find?
Sure. So this is part of the soybean group called Science for Success. And so it's funded by the Soybean Checkoff here. So, we try to do research that's impactful to farmers, and we were getting together and noting a lot of farmers in all these states had the same questions, and they call these products very broadly biologicals. We're looking at living microorganisms, which we would call biostimulants, but a lot of questions were coming in for farmers. So together we developed a protocol where we could evaluate some of these biostimulant products across the US. And so we were able to do that for 2 years. Not every product every year, because the turnover is so high, and not everything is registered in every state either. So there's some complications there with multi-state, multi-year research. But overall, we were able to evaluate products at 104 different locations across the U.S., which is really a phenomenal data set.
Yeah, and so is that funded? You said the soybean checkoff, but is that the national checkoff that funds that, or is that each state?
So the Science for Success team is funded by the United Soybean Board, which is the national checkoff. And then this research in general was funded by a co-funding opportunity with USDA and United Soybean Board.
Okay, cool. So walk us through, like, how many products did you look at? What did you, maybe we can go, Jay, to Laura's first slide, and that would be a good place to start.
Yeah, well, that was our starting point with our question because farmers were really confused. And it was because this is an extremely confusing space. A lot of these products are called biologicals broadly, but you have biopesticides, microbials, non-living ones, living ones. So a lot of confusion. We focused on, again, those biostimulants that were living microorganisms, and we evaluated 9 products that were commercially available at that time each year. They weren't the same each year, but for the most part, they were the same each year. There were a few turnover products, but we had 9 each year, and we had our untreated control, and our untreated control was treated with fungicide and insecticide. And so we put the biological treatments on top of that fungicide and insecticide, which we had to check with each company to make sure they were compatible, because we didn't want to have any issues there. But we had our non-treated check, which had fungicide, insecticide, and then those 9 biostimulant seed treatment products for 2 years.
Yeah, and that is a really key piece of that thought process. And Greg, you mentioned endophytes, and as soon as you go to endophytes, can you put them in chlorinated water, right? Like, the endophytes have the exact same problem of where you can put them and not, correct?
Yeah, absolutely. Sensitivity around how you deliver the microbe, the biostimulant, to the plant is probably concern number 1 or 2 on anyone's list.
Yeah, particularly with a living product, right?
Right. And I think, is there a slide that actually shows the microbial strains, Laura?
I don't have a slide with that. It's on our Science for Success fact sheet. I don't know if there's a way to drop it in the comment box. But yeah, and we don't have the company names listed. We just have the active ingredients as the microbes listed.
Right. But all microbes and all seed treatments. That was the focus for the trial, if I heard you right?
Yes. Yep. They were all seed applied, and they were all living microorganisms. That's correct. Yep.
Yeah. Very cool.
So maybe we'll go to slide 2, and Laura, you can kind of continue with your story, because it's just a fascinating story.
Yeah, so we were able, I think, to get somewhere between 25 states or so involved, 104 environments. You can kind of see them here. And of course, there's a lot of diversity in crop rotations and soils and weather. So those environments we clustered, and those clusters are based on soil factors and weather factors. So you can see the different colors and different symbols are the colors. But I mean, we went all the way down to Florida, up to North Dakota, into Nebraska, all along the East Coast. So a very, very robust data set.
And not the same sites every year, just looking at the two slides, or the 2 years rather?
Yeah. We had some states do it one year but not the other. For example, Iowa, they had a site in 2022, but then 2023 they flooded, and so they had no sites. And then in 2023 we had some new agronomists jump on board. They were brand new, hired, and they started the project in 2023. So there was consistency, but slight differences between the 2 years. And then the fields themselves were different each year because most of
Right.
these, in the Midwest at least, were corn soybean rotation, so different fields each year.
And so the clusters were, though, weather related, climate related.
Yes, weather, soil related. Yep.
Yeah. And so did you see in the data any difference between the 4 different climate clusters?
Yeah, mine yielded the highest, those blue ones. So, which is probably not a surprise. The U.S. Midwest is very productive, so we had our higher yielding sites there, and then also going up to like Wisconsin, Minnesota. Those red circles up there were pretty good yielding. But you get down to Louisiana and Florida, especially some of the lower yielding sites. So there were yield differences you could see
Yeah. Cool.
in
Cool.
the areas.
Yeah. Okay. And so then carry on with the story. You maybe go to slide 3 if you could, Jay.
Yeah, so this was a huge data set with all of these states, 104 environments, and we also had each location do 6 replications. So we knew that we would need a lot of replications to try to tease out significant differences. So we had 6 replications at each site. So we worked with a statistician, and so he looked at the best product at each location compared to the non-treated control, which is a big assumption because the best can vary across sites. But he picked the best at each location and compared it to the non-treated control. And the best he could do was about 3 bushels per acre, 25% of the time. So if you could pick the best product for your location compared to the non-treated control, 3 bushels per acre, 25% of the time.
So explain that a little bit more. So if I looked, could I get to 50% of the time and gain a bushel and a half? Or how does that data shake out?
Yeah, that's a great question. So this is a very hypothetical scenario since we can't pick the best one. But yeah, the story is that there was no product that consistently improved yield across the U.S. And 3 bushels is a pretty large yield increase. We did some power analysis, but yeah, 3 bushels, 25% of the time was the best we could do, and no product consistently improved yield across the locations.
So you got to tell me, because what's a power analysis?
Oh gosh, that's another statistician question.
Okay.
But no, so basically we wanted to see at these different yield thresholds how many locations we would need to actually get statistical significant difference. So 25% of the time is not what we would usually call statistically significant. And then not even usually, it's not statistically significant. So we looked at an 80% threshold, and for a 2 bushel per acre yield difference between a product and a control, we figured we would need, we figured, the statistician, about 25 to 30 different experiments to get a really small yield difference, maybe half bushel per acre. We would need 200 experiments to tease out those differences. So I think one challenge in this space is these biological treatments may help improve yield, but those yields can be small, and you just really need a very, very large data set to tease out any potential differences.
Yeah, interesting. But you had 104, right?
We had 104. We had 104 and couldn't tease out those differences. Yeah.
Yeah. So when you say you'd need 20 or 30 for a 2 bushel difference, that you should have found that if it was there.
You should have found it if it was there. Yes, absolutely.
Yep. Yep. And so the next question, and I think your slide 4 really addresses it, is, are there certain environments, or like how do we pick from those and from that data, or try to pick the right living biological?
Yeah. Yeah, so I always try to think about, these biologicals should be a symbiotic relationship between the microbe and the plant. And so I try to, when I talk to farmers, compare that to the disease triangle. And I don't know if you guys use that as
Oh,
much as
yeah.
we use
Yep.
it. Okay. Well, I always tell, make the joke at our extension meetings that every plant pathologist is contractually obligated to show the disease triangle at every meeting. But with the disease, you have to have three things in place at the same time. You have to have susceptible host, the virulent pathogen, and the conducive environment. And if all those three things don't come together, you don't have a disease. And so, similar to the biostimulant products, you need to have a host. So the soybean plant, you have to have the plant beneficial microbe, and you have to have the conducive environment. And so, with the plant beneficial microbes, you have to have them, but you also have to have them alive. You have to have them alive, and you have to have them in a high enough concentration. And then you're putting them into this environment, the soil that already has a ton of microorganisms. So I think there's a lot of places where this could potentially break down to maybe not having a yield increase or that symbiotic relationship, because those three pieces really have to come together, and there's a lot of challenges to do that.
Yep. And so I'm really intrigued because the plant or the other microorganisms in the soil would be a huge bank of competition for what you're putting in. And I think Greg's number, when I chatted to him on Friday, was like 6 billion other organisms we have to outcompete. Did you think about taking those 10 different bacteriums or organisms that you and put it into a sterile environment in a lab somewhere to see if you could get any impact by doing that? Laura, did you try that in the lab at all? Yeah, yeah.
Yeah, sorry, I thought you were talking to Greg.
No, sorry.
We
No.
Yeah, no worries. So we did not in this larger data set, but some colleagues at Michigan State looked at the greenhouse, not a lab setting, and so they tested some of these in a greenhouse setting where they could control the environment more, have sterile soil, things like that, and they withheld water, and so they did see some of the products, help with water relationships. It was less wilty. The plants were less wilty when they had the microorganisms. So I think the environment, the soil environment makes a really big difference, because they can see some of that in the greenhouse.
Yeah, yeah. And so just Dave in the chat is asking, they were all in furrow or seed treatments, but these were all seed treatments, correct?
All seed treatments. Yep.
Yeah, all on the seed, and so right with the seed. And Mark, you're right. Yield really doesn't matter. It's return on investment. But Mark, if you don't get a yield increase, she's pretty tough to get return on investment. So that's sort of I think the game that we're playing right now. So Greg and Jay, that's probably good on the slides for now. Greg, jump in here and do we just walk away from live organisms in the soil?
Yeah, that's a good question, and I guess I just wonder whether live organisms in the soil is maybe one of the more difficult places to get traction in the whole realm of biostimulants. Again, I don't really have a horse in the race, but in the whole realm of biostimulants, I might be more interested in applying something to the foliarly to the plant where I eliminate some of that competition and getting swamped by other microbes in the in furrow environment. And even sometimes I get concerned about the length of time that exists. Well, soybeans are a little quicker, but when we think about planting corn on the 25th of April, how many days do we go in Ontario before we have any root tissue that there could be some sort of interaction between the microbe and the root? And so I get concerned that maybe the in-furrow environment is a bit of an uphill climb, right? And I guess
Yeah.
I had one question for you, Laura, and that would be, how many of these were just straight up rhizobia inoculants that we would normally use on soybeans as, just because we want to ensure that we have good nodulation.
Yeah, so there was one product that was Brady Rhizobium japonicum, and then these were all products that could be sold to a farmer. So sometimes in these products you might have 4, 5, 6 different microbes, and one of them maybe Brady Rhizobium. But we tried to look at other things since there's been a lot of research on Brady Rhizobium japonicum, but they were thrown in as those, I don't know, premixes. I don't know if that's the right word for it, but in the jug.
Yeah.
Right.
And all of these had soybeans before, so you would not expect to see big yield increases.
That is correct. Yeah, these were all traditionally in that corn, soybean, wheat. If you go south, maybe some cotton in there, but they all had soybean in the rotation. Mm-hmm.
Yeah.
Do you have a number? If a grower asks you in Ohio, what's the value of inoculating even in fields that have had long histories of soybeans? Do you get that question? What is my value of a Brady Rhizobium inoculant even in long-term bean fields?
Yeah,
And
so I got that
what's
question.
your answer?
Yeah, I got that question a lot when I first started because my predecessor always promoted Brady Rhizobium every year, no matter what. And he always said there was a 3 bushel yield increase. And
Wow.
I figured out he was using a different p-value than me, right? And so he was using like a 70% confidence level, and so there was a yield increase if you have this 70% confidence level. If you raise it, usually for statistics and for publications, usually we have to be 90 or 95%, and if you raise it up there, then there's no yield differences. So
interesting. 70%. That's Peter Johnson. That's Peter Johnson statistics almost now.
Yeah.
No, just because we get that question a fair bit, and I think our answer in Ontario, I think Horst Bonner's number in Ontario is a bushel for inoculating even in long-term bean fields. Not 3 for sure, right?
Sure. Yeah.
Yeah.
Yeah. And, this study, we didn't see that. This was the historical when I reanalyzed that data set. Yep.
Yeah. And Greg, I concur. It is 1 bushel that Horst talks about. And I don't know, there's lots of people who have tried it and just can't even find that bushel. And so they quit doing it.
Sure.
And other people just keep doing it because they think they should. So before we change gears, because we should change gears, Jay, maybe it's time for our first read if we could. And we keep our sponsors front and center.
Our sponsors tonight are Profitable Practices, MNP, and the Wheat School. From varietal advancements, protein and yield management, precision farming, and on to marketing, Real Agriculture's Wheat School is a video series that tackles every facet of the wheat growing season in an engaging and informative format. The Wheat School is made possible through C&M Seeds and Bayer Crop Science. Learn more at wheatschool.com.
Yeah, there we at least get one good sponsorship tonight. Wheat, it's all good. And so I see Dave put in the chat, and I think it's pretty funny on the inoculant thing. He's saying 60% of the time it works every time.
Yep.
I like that. That's kind of a bit of fun. So Greg, if we agree that it's probably the toughest spot to make a biological work is your category of soil-placed living organisms. Let's move to the next category. Do we have a shot with plant extract soil applied, or is there still too much pull there? What's your thought process?
Well, I find it really intriguing that when we talk about what you can put in furrow to stimulate, the interesting thing is that I don't believe these products are mutually exclusive. I think about it. So at Syngenta, we've got a product that is an in-furrow biostimulant, but it has no living organisms in it whatsoever, right? It's sort of a little bit of humic acid and some proteins and some betaines and some polysaccharides, try to stimulate some of the activity right in around the root zone. And you wonder, from the other camp, if I don't have the right microbes in the right populations, what am I trying to stimulate? And so I get really intrigued by, should I be focusing on microbes, or should I be focusing on stimulation of just whatever is naturally occurring in the root zone? And I guess with the overarching idea, hope maybe is a better word, that that there are things that can happen in the root zone that prime the plant for future success, and that we don't necessarily think that everything that has to happen in this root zone for whatever biostimulant or biological, that might not have a lingering effect, but can you prime that plant early to get it on a yield trajectory that's higher? And that still excites me about what could happen in furrow to change the trajectory of the crop from a yield perspective.
So it excites you, but do we have a good product or good science that actually we can point to say, yes, that's real?
Yeah, well, I'm sure there are lots of private companies that would absolutely answer that question, yes. And in my own research, we have some instances where, mostly dominated by low testing soils, like low soils that are low P and low K, where we get a stimulative effect to putting some sort of plant extract, humic acid, biostimulant. Again, I'm in the non-living microbe world, but when we go into some low P and K environments, that we get a bit of an uptick or a priming effect from those biostimulants. And that's intriguing, but it happens right now at much too low a percentage of the time to get overly excited about it. Excited in the sense that it looks like low P and K or tougher soil environments are more conducive to a positive response, but not at a frequency now that we would be able to recommend it across, a wide range of soils. And I don't know, to go back just a little bit, Laura, was there any indication? I'm assuming not, because the data was extensive and it was hard to pull out positive responses, but was there any indication that tougher soil, lower P and K, I don't know, Florida, Georgia, sands, I don't know, I guess that you saw some sort of hope?
Yeah, I mean, we hypothesized that, maybe further south where the soils are more weathered, there'd be more there. And that was part of the reason with the different regions and such that we could tease that out. But we weren't able to tease that out. We did do some initial analysis because there were yield differences in the regions. And so, again, we worked with a statistician to figure out, okay, the biostimulants did not influence yield. Can we, see what did? So they ran some models. And so the things that influenced yield were things like row width, crop rotation, seeding rate, planting date. So those traditional agronomic things are really what explain yield differences in the data set.
Yeah, fair enough.
Yeah.
But we shouldn't say we were hoping. You shouldn't hope for things in research, but when you do this large study, you really hope that you can, have a recommendation you can hang your hat on for a farmer. If you have sandy soils and, this region, weathered soils, this biological treatment would be great for you. That's our hope, is always to have recommendations for farmers, and really it kind of fell back to the traditional agronomic practices that influenced yield.
And I'm quite sure that the soybeans yielded the most if they followed a wheat crop.
Yes. Yes, we love it. We have data on that. I like to call it, this is off topic, I like to call wheat the better biological, right? So when we have wheat in rotation.
Oh my gosh.
When we have.
Okay. It's 8:32. It's 8:32, and the world has come to an end.
5 bushels on your soybean yield. 5 bushels if you have wheat in rotation, and that is statistically significant. So, and it is biological. Yeah.
I'm quoting you. Like you are going to become world famous if I can make you
All
world
right.
famous, Laura. With that quote, man, I love it. That's awesome.
Wheat is the better biological.
Yeah, weed is the better biological. That might become the quote on the bottom of my emails now. That's such a good statement.
Take it. Yep.
Gosh. And so Gord actually asked, I think, Greg, essentially your question, right? And Mark is in the chat. And one thing that has always surprised me, Mark's on very heavy clay and has lots of hog manure, so very good fertility. But Mark often gets response to things like better response than what we would typically get other places. And so, Laura, is that like, are all your research sites kind of the best soil? Oftentimes when we go on station, it's the flat, beautiful soil. It's not the stress soil. Do you have sites in that 104 data set that would really fit that tougher category?
Probably not quite as much, right? So ours are on farm. The ones in Ohio were on farm, but we do it with our variety testing program because we're there and we can put things just right off to the side of a variety testing program. So when we do variety testing, we want farmers who are doing a good job. Granted, weather gets in the way, soils get in the way sometimes. We have heavy clay soils as well, but these are sites that should have adequate P, adequate K, decent drainage, good pH. The other states, there are soils that are by nature just more poor quality in other states. I think Florida was the lowest yielding. I think they were about 20 bushels per acre, where, we go to Ohio, sometimes we can get, 80, 90 bushels per acre in a good year. So there were just more soils that were naturally not as productive, but I would say it's probably fair to say most of these were high yielding soils, either managed well by a farmer or managed well at a research station.
Right. Yeah. But you would, like in Mark's case, and Mark put in the chat there that, an unbelievably good result last season. They were in extreme drought. They had, soybeans that yielded as low as 8 bushels per acre and corn that was yielding 40 bushels because they just had no moisture. They're not really deep soils, no moisture. And an abiotic stress reliever that they were trialing got, really good results. But then again, Greg, to come back to your point, that's a really stressful environment. And I doubt, and I don't know, Mark, whether that was in furrow, but I doubt it was in furrow. It was probably more applied foliar. So Greg, like, are there any instances that you can show us? Like what about your slide 1 and weed control? Is that a signaling thing from a stress reliever, or walk us through this, because this is interesting.
Well, again, it's a little bit, unfortunately, underlines the hope idea, but there is a bit of science in it as well. And so, in Ontario, we're very familiar with Clarence Swanton's weed research. And when you look at that picture, you say there's interference that comes from that weed competition. And the interference is not a lack of water or a lack of sunshine, or could be fixed with any amount of N, P, or K. Those are all non-limiting in these situations. So you have this interference that is really a signaling within the plant. And even when you come in and wipe all those weeds out, and it's clean from June the 14th or 24th on, you still have a legacy effect that the interference caused. And so we're all familiar with that. And I liken this priming effect, or this signaling effect, that if you could stimulate the roots. So I have a picture here. The lower roots do have an in-furrow biostimulant. There is some evidence that we get some root proliferation. If you dig the plants up, you see that the soil seems to adhere to the roots better under the treated block than the untreated block. But again, I'm of the opinion that these biostimulants in furrow, in this situation, as you need to expect that what they can do is prime the plant to get them on a better yield trajectory. And I guess if we really wanted to chase that, we would say, is there a foliar biostimulant that you could have sprayed on the corn plants on the left to overcome some of that signaling that reduced the yield trajectory. Well, we've tried that. We don't necessarily have any answers there, but at least I like to position this as where we need to think about biostimulants. There's not an NPK, moisture, sunlight solution. This has to be considered a bio signaling, and that gives me some hope that if you can signal within the plant, you can get it off on a better trajectory. And this is a picture that captures some of that idea in terms of an in-furrow root stimulator.
And that signaling is really interesting, right? And Laura, you're well familiar. You did your PhD on some of this, but it's that the plant can see, if you will, that signal even before it emerges or just as it's emerging, and that light signaling can have such a big impact on the plant. Like, is there a way that we could find something to prevent that plant from having the impact that we clearly see when we get that root change? Is that even possible? What do you think, Laura?
Oh, anything is possible with science, right? Well, I think biologicals in general, there's a lot of excitement in the future. I think there's a lot of development that's going on. There's a lot of ideas. I think that, anything is possible. It's not my area at all. But, plants, there is signaling, and there are, I don't want to say the plants are talking to each other, but there is signaling, and they do change how the plants grow. So, I think there could be additional steps where something like that could happen.
So, yeah, really interesting. I interviewed Clarence Swanton for a Real Ag video a week ago or something like that, maybe 2 weeks, and we will be putting that video up at some point. But and I don't think it's in the video, but when we chatted, he actually said there's a researcher, I believe in Minnesota, who thinks he's found a soybean genetics that is that does not get impacted by that light reaction, that it's essentially resistant to that signal so that it doesn't change how it grows, and those early weeds don't reduce the yield. And I mean, they haven't got totally down that road, but if we can find that genetically, it's probably possible to find some form of a biostimulant or product that could change that signaling as well. And it would be really exciting if we could only find it. But so far, Greg, we haven't found it, right? At least not in your experience.
Yeah, but it certainly keeps our eyes wide open that there are things happening in the plants that are very subtle and are related to plant growth regulators or biostimulants or very subtle communication up and down the plant. And why can't we play in that space? We know it doesn't take 100 pounds of nitrogen to change that communication. It takes very small amounts of biostimulant to impact that, if you could find the right chemical pathway.
Yeah, if you could find the right chemical pathway. And so I do see that Mark said what his his good results last season from extreme drought, it was weed control. He sprayed Acuron and killed the weeds. So yeah, Mark, that it doesn't quite count as a biostimulant. Who knows what it does in the corn plant, but we know what happens when we kill the weeds. On that one, I think on that note,
thumbs
we are
up
going
for
to
Acuron.
Yeah, right. Syngenta products. So Greg's in, right? Jay, let's go to our last read of the night, and then we'll shift gears again.
Our sponsors tonight are MNP, the Wheat School, and Profitable Practices. Profitable Practices on Real Agriculture is a video series featuring Canadian producers who are adopting farm practices to have a positive impact on profit, people, and the planet. Profitable Practices is made possible by support from Farm Credit Canada and RBC Royal Bank. Find out more at realagriculture.com/profitable-practices.
Awesome. So I may I see. Mark says it was a product that was tank mixed with the Acuron. It wasn't actually the Acuron. And so field trials, 120 feet long, over 3 farms, that, maybe there is something there under those really extreme drought conditions. So just from that perspective, and stress, Greg, maybe we'll go to your slide 3 and let's talk a little bit about how we might have an impact under stress conditions, right?
Yeah, so this is nearby grower here in southwestern Ontario, where we're using Megafol. So Megafol is our plant extract biostimulant. And there was a question earlier, when I say plant extract, do I include algae and seaweed, et cetera? And of course I do, right? And so in this particular situation, in 2023, we saw a nice yield bump for putting a half liter of Megafold down with the T1 and the T3 applications in winter wheat. So a 147 to a 155, again, fairly consistent in the field. And that year, the May 10 to June 10 rainfall was a whopping total of 16.7 millimeters, right? So that's not much rain for a pretty critical period in wheat growth, May 10 to June 10, right? So we fast forward to the next year, and I got essentially no response to my half liter of Megafol going down at T1 and then again at T3. And when you look at the rainfall that year, 78.9 millimeters. So it sort of gets back to Mark's comments about the significant yield response when you get stress conditions that rise to a certain level, right? And if you say your product is abiotic stress related and you have very little abiotic stress, maybe it's not so difficult to believe that, well, maybe we shouldn't have expected too much yield out of that product. But when you certainly move to the 2023 data, yeah, we had stress. I could not get my soil probe into the wheat field that year. It was so hard and dry.
And I can't argue with 155 bushel per acre wheat. Like that's, I don't know. I don't even think Laura could argue with that kind of a wheat yield. That's pretty spectacular. But so Greg, that's one site. How consistent was it? Because 2023 was dry, lots of places.
Well, our 2023 number is a little more robust than our average biostimulant increase. We're 2.5 bushels on average, and multi-year average. And in the 2023, it would have ticked up over that, but maybe 4 sort of bushels per acre improvement. So it does tick up in the 2023 stress. I would love to say that it was, always 8 or 10 bushel everywhere we went in 2023 because of the dry conditions. But about all I can tell you is there was a significant uptick in response to Megafol in the dry year, but, isn't as consistent even across a dry year as we'd hope for. But in the world of people that are interested in biostimulants and sort of keen to make them part of their operation, the trend is, you get the product, you get it in your shed, and you play the stress game, right? You look for the opportunities to combat stress.
And so Laura, like I was going through some of your eFields data, and there's some biological trials in your eFields data. Have you looked at all at these foliar stress relievers and tried to tie it back to either heat stress or drought stress, and any thought process or yield numbers?
Yeah, so I'm not a huge player in e-fields. I look at it here and
Yeah,
there, but not a huge contributor to e-fields. But it's a great on farm program. Elizabeth Hawkins is the main person there, and I've heard her talk about biologicals when I give my presentation. And we come at it from a different angle of on-farm testing versus, maybe some more small plot type things, but the conclusion is the same. She doesn't see a whole lot either, which is good for farmers to hear a similar message, but from two different angles. And I know we've had some pretty severe droughts in 2024 and 2025 in Ohio. We had a D4 drought for the first time in 2024. I don't know if she had research locations down there. I haven't teased that out, but overall, she hasn't seen a lot of response with the biological products on farm.
Yeah. And so, Greg, can you help us predict? And again, Mark's in the chat. Mark's one of those guys that gets response to Megafol. When I work with you, Greg, and we go to Syngenta on that beautiful silt loam soil at Syngenta, it just seems like it never goes under stress, and so it's really tough to pull out any yield benefit. So, do we know when are different crops different? I've heard you talk about potatoes and endophytes, how nitrogen fixing endophytes seem to be work better on potatoes than it does on any real crop like wheat. Just can you give us some kind of guideline? Because Margaret's in the chat and she says, so we call them refined reptilian extracts, which is a quote from Nick Stockman at Strathroy, a farmer, Laura Lindsey here in Ontario. So like can we pick those conditions out?
Yeah. Well, I think that's where we're at right now in biologicals, is actually trying to identify the stress by the crop, then by the biostimulant. So for example, I put Megafol on corn in a non-stress year, and I get maybe 3 or 4 bushels, and probably not statistically significant. I pull out my weed whacker and defoliate those plots, right? Give them all a haircut with my weed whacker, and then spray the Megafol, so my simulated hail project, and I get 8, 9, 10 bushels fairly consistently when you've stressed the crop, right? Out west, my colleague Chris Dummigan has looked at stress on lentils, and he can see an advantage to Megafol, and the driver is the number of days in the growing season that's over 30 degrees C. Now, I think that's where we're at. Over 30 degrees C on corn in Ontario might not trigger anything, but over 30 degrees C in certain crops in Saskatchewan, that might be a trigger that a biostimulant actually has a significant play. And so I think that's where we're at right now, is trying to categorize stresses and which stresses impact which crop, and where the biostimulant opportunity might be.
Yeah,
it would be nice if it was simpler than that, but I'm not sure that it's going to get simpler than that.
Yeah, and so I asked Jay to pull up your slide 4, and that's your heat stress slide, right, Greg?
Yeah, so exactly. You've got along the x-axis, you have days over 30 degrees, and then you've got yield improvement up the y-axis, and so you get this, I don't know, sort of a consideration, at least, that as the heat intensity increases, your odds of getting your ROI on a biostimulant, go up, right? And so that's probably as real as we're gonna get it, in at least in the near term, until we crack some of those things like we talked about earlier, which is what is the exact chemical signal that you're trying to combat in a heat stressed lentil field. Well, we don't necessarily have those answers, but at least we have examples where a heat stress field responds more positively to the biostimulant.
Yeah, like that chart, when I look at the data, when there isn't many days, there's quite a bit of chatter in the data, right, Greg? Which is sort of what we often see. But when we get out to quite a few days, then that is a little bit more of a relationship. So I don't know, Laura, does that make sense to you to try and find those products or those triggers?
Yeah, I think it's an interesting way to look at it, and that graph I think is a nice way to potentially present that type of data. And yeah, I think it's interesting. And we've done some lit review on biologicals as part of this work, and countries that do have stressful conditions, so do tend to see more positive yield response to biostimulants. So yeah.
And so you're talking specifically about that project that your grad student did on and compared Brazil to the U.S., correct?
Yeah, that, and other countries as well. I think
Okay.
Iran or Iraq, Middle Eastern states or countries may see more response. But yeah, I had a student who did a literature review to compare biological products in the U.S. versus Brazil, because he was from Brazil, and he saw better efficacy of products, biostimulant products in Brazil compared to the U.S., which would be more weathered soils, probably more extreme environmental conditions in some cases, or a lot of cases, and there was more efficacy. And so we don't know if it's necessarily because of the soils and the weather and things like that, because there's a lot more oversight and regulation in Brazil compared to the U.S. So maybe there's part policy piece, part environmental piece, but I think weather conditions and growing environment definitely plays a role.
Yeah. Yeah. Interesting. And so the trick though, Greg, that you have to develop for Laura and I is on each crop, what is that trigger? And in corn last year, they talked about it got too hot during the night, and that's what kind of slowed down corn development, that it was just really slow because of very hot nights. So I need to know what to spray when that happens. Uh, like the trick is, can we predict the stress, and then we know what product to use if we can predict the stress?
Right. So this is the request across the landscape, is we don't want to let me act like a little bit of an idiot, but we don't want to put seaweed. We don't want to put seaweed in the blender and then throw it in a tank and hope that something's going to happen, right? We want to take seaweed and carefully extract the key components out of that seaweed, and then apply it to crops and see what genetic impact that extract could have on a plant. Does it actually upregulate genes that reduce water loss in a drought? So until we get the science figured out, that we go to the plant, we do the extract, we take the extract, we apply it to a crop, and then we assess what changes are happening within that plant. That's what's going on now in the background, right? And we need that to be developing, so that, well, new products, right?
Yeah, and I think that sounds like a wonderful PhD project. And I'm pretty sure if you would do that PhD project, Greg, that Laura would be happy to be your advisor. And so, away we go. Because I, I,
I'm in the position of sort of chalking it up that I have failed at biological biological effort, and move on, leaving a trail of suggestions in my wake.
and so we have about 4 minutes left. Greg, can you just quickly, because you're a corn guy, is there anything we can do? We've talked lots about kernel mass, about keeping kernels on the cob. Any thought process that something we might be able to do from a biological there?
Well, it's a terribly interesting story how corn yields have improved over the last 50 years, right? And when we look at Tony Vine's work out of Purdue, we see that about 2/3 of that corn yield improvement comes from heavier kernels, more massive kernels. And so I look at that and say, wow, we increase the density, and we try to push for more yield. And what goes on within the plant that would support more yield? So historically, what supports more yield has been just the genetics. I mean, the genetics, hands down, have supported yield more importantly than anything else, right? But we go to fungicides and we look at when we try to push density a little bit in hybrids, modern hybrids, they often respond to a fungicide to hang on to more yield than even they would have without the fungicide, right? And so I think that's a really another interesting place. Can you get a biological that would improve the flow of photosynthate from the leaf to the kernel and promote heavier kernels? The genetics are already wanting to go there. The fungicides, I think, mostly support that idea of holding on to canopy greenness, extending the grain fill period, and therefore producing heavier kernels. I think that's a really exciting option for biologicals to be able to stimulate better movement of starch from the leaf to the kernel, and that's intriguing. And so I know I've said that a lot tonight about it's intriguing, but we have some products in the space now that at least have that sort of target of upregulating phloem loading so that it gives you heavier kernels. So I think that's and if you're looking for an experiment to do on your farm this year, to me that's a really exciting one. Bump the population, from 34 to 38,000. That applies some additional stress to the crop. It struggles to hang on to ear size at 38,000 plants per acre. And then look at your late nitrogen, your fungicides, or heaven forbid, a biological that might play in that space of hanging on to ear characteristics and yield as you push those modern hybrids. To me, that's a really fun place to look.
And so just to wrap it up, would you choose a plant growth regulator in that situation? Would you pick a plant extract in that situation? Or would you pick humic acid? Which one would you pick to do that? And I'll ask you the same question, Laura.
You got to say PGR.
Sorry, PGRs. Yeah, plant growth regulator. That was my first one. Yep.
Okay, sorry, I missed that. Yeah, I'm going to sound like a really well-rounded salesman here and suggest that that I'm in the camp of probably a plant extract, right? Compound, or maybe a PGR. I think those would be the two that would excite me the most about trying to upregulate the grain fill. And some of the more traditional agronomists would say, well, if you don't have enough nitrogen down, and if your potash level isn't at 160 parts per million, and et cetera, or you haven't picked the right hybrid, and I would have to agree to all of those after you've done all of that very basic important stuff, then maybe there's an opportunity for a biostimulant plant extract or a PGR to maybe play in that space of improving kernel mass.
Yeah. And so, Laura, what's your thought on that? And in addition, I think Jason asks an excellent question. What do you think the future of biologicals is?
I'll answer his first. So the future of biologicals, I think, I've talked to a lot of these companies, and there's a lot in development. So I talked to all of them that we've tested, and some of them are shifting away from actual microbes to how to deliver them. And so instead of developing microbes and new strains and things like that, they're working on packaging. How do you package them? How do you apply them? How do you deliver them to keep them alive? So I think that's a really interesting part of this as well, not just the micro products, but how do we deliver them and store them and things like that. So that's, I think, an interesting development that industry is working on. And then your question, Peter, I'm going to talk about soybeans instead of corn because we consider corn a rotational crop in Ohio for soybeans. So soybeans, I'm going to get away from products and talk about more of my soapbox. I think, honestly, planting date is the best way to maximize soybean yield and get heavier kernels. We've done a lot with planting date in Ohio and trying to push it earlier. And what we're seeing, when we plant soybeans earlier, we get a really long flowering period. And so I'm really interested in this earlier planting, earlier flowering, longer flowering into, the growing season, and really using that to mitigate stress, because soybeans can take those short periods a lot easier, better than corn, because it can flower for such a long time. And then, we do have pockets of dry weather here and there, but if you have flowering for such a long period of time, I think you can make a lot of gains there.
Yeah, interesting. So that's a whole nother
It's a whole
discussion.
other thing, because I'm not going to say PGRs or fungicides or necessarily products, but planting day is one that I think where we've seen a lot of gains in the soybean world.
And we have talked planting date on the agronomists a couple of times, but your comment about a longer flowering period with
Yes.
earlier planting, that I'm going to have to chase Horst on that, Greg, and see if he has that similar data, because that's really, really intriguing. Cool. Well, we are out of time.
I would like to throw in one thought, is that for those of us, and I get accused of sort of having jumped into the snake oil world, et cetera, et cetera. I would like to say that those of us who are working on biologicals, at least from my perspective, we appreciate, I relish the data set that someone like Dr. Lindsey produces to keep us honest. I do not want to sell stuff that doesn't work. So I personally relish data sets and professional sort of keeping us honest. I think that's key, and we'll keep trying and we'll keep asking good questions, and a lot of it's going to be unsuccessful, but we're not afraid. I'm not afraid of having excellent data like Laura presents today to try to keep us on the right track.
Yeah,
I appreciate that.
And one thing about you, Greg, you have very high moral standards from that perspective, and that's. But it is good, Laura, that you and the whole soybean group do that. And as you said, it's too bad that you can't say, yes, this product works, under this condition.
That's what we really wanted, but we can only say what the data says, right? Yeah.
Right. Yeah. Don't say the data is wrong. The data doesn't lie.
Yeah, the data is the data, and it's not every product, everything it's such a large area, but we can just speak to what the data shows.
Yeah, no, absolutely. No, an excellent night tonight with my best quote of the year. Wheat is the better biological. I couldn't be more stoked. Thank you very much, Dr. Laura Lindsey from The Ohio State University and Greg Stewart from Syngenta. An awesome night. Thanks for everyone in the chat for the questions. Have a great night, everybody. We'll be back next week. Oh, don't forget CEU credits. Just send us an email tomorrow. Thanks very much. Good night.
Thank you.
Bye.